Hello everyone, welcome to today's live broadcast, New USP 1058 Analytical Instrument Qualification Regulations, what the 2017 requirements mean and how to comply. I'm Ali Matheson, the Editor-in-Chief of LCGC Europe and the Column, and I will be your moderator for today's event. We are very pleased to bring you this web seminar presented by LCGC and sponsored by Agilent Technologies. Agilent is a leader in life sciences, diagnostics and applied chemical markets. The company provides laboratories worldwide with instruments, services, consumables, applications and expertise, enabling customers to gain the insights they seek. Agilent's expertise and trusted collaboration gives customers the highest confidence in Agilent's solution. Information about Agilent is available at www.agilent.com. We have a few important announcements before we begin. This webcast is designed to be interactive and we encourage you to ask questions during the event. You can submit questions by typing them in the Q&A box, which can be found on the right-hand side of your screen. You can move, minimize or enlarge the slide window at any time by clicking on the icons in the upper right-hand corner of the slide window, or by hovering your mouse over the lower right-hand corner and dragging the window to the desired size. The slides will advance automatically throughout the event. If you have any technical problems viewing or hearing this presentation, please click on the question mark help widget in the dock at the bottom of your presentation window. Please also note the resources provided by our sponsor, which you can locate in the green resource widget located in the dock at the bottom of your screen. Concluding today's event, we invite you to take part in a very brief survey from our sponsor. The survey will automatically appear on your screen. I would now like to introduce today's speakers. We are very pleased to be joined today by Dr. Bob McDowell and Paul Smith. An analytical chemist with over 45 years experience, including 15 years working in the pharmaceutical industry, Bob McDowell has been a consultant for 25 years. He has been involved with the validation of computerized systems for over 30 years and has recently published the second edition of Validation of Chromatography Data Systems. Bob is the author of Questions of Quality in LCGC Europe and Focus on Quality in Spectroscopy, as a member of the editorial advisory boards of several journals. He was a contributor to the GAMP Good Practice Guide for Validation of Laboratory Systems, second edition, and initiated changes to 1058 through publishing the stimulus of the revision paper and draft revisions of 1058. Paul Smith started his career as an infrared spectroscopist and held a number of quality and management roles in the 17 years he worked in the pharmaceutical industry across quality control, new product introduction, and research and development. For the past 12 years, Paul has worked in the laboratory compliance consultancy roles. Paul monitors regulatory compliance trends and shares the information through Agilent seminars and events. He was a contributor to the GAMP Good Practice Guide for the Validation of Laboratory Systems, second edition, and a regular contributor on Analytical Instrument Qualification. And thank you, Bob and Paul, for joining us today. And if you could start the presentation, that would be great.
Good morning, everyone. I'm Bob McDowell, and I'm going to be giving the first part of this seminar. And I want to cover essentially three parts, or there's going to be three parts. I will look at in part A, the significance of the changes in the new version of USP 1058. I'll pass over to Paul, looking at the role of AIQ and in data integrity, and then I'll come on and finish off with looking at how to comply with the new USP 1058 requirements. And then we'll finish up with a session on questions. So let's start off looking at the significance of the new changes to USP 1058. For those people that know the old version from 2008, it may look fairly similar, but there are some substantial changes in there, and we're going to go through them in this section. But let's start from basics to begin with. Firstly, the United States Pharmacopoeia is the only pharmacopoeia that has a general chapter on analytical instrument qualification. So it is a very important general chapter from a global perspective. The numbering system of the USP means that as it's over 1,000, it is an informational chapter which is very strong guidance rather than a mandatory chapter that's under 1,000. But the key thing here is that the best practice is outlined in USP 1058, and the individual instrument requirements for qualification are highlighted in the individual general chapters under 1,000 for each analytical technique. USP 1058 was first published in 2008 and has been untouched for 9 years. Although the USP through the American Association of Pharmaceutical Sciences had a review meeting in New Orleans in 2010, and that was co-chaired by Paul, who kindly invited me to give a European perspective at that meeting. Coming out of that was an agreement between Chris Burgess and myself and the USP to write a stimulus to the revision paper that was published in January of 2012. Following feedback from that stimulus paper in Pharmacopeal Forum, we went through and drafted a more detailed revision to 1058 that was submitted in 2013 to the USP. After revision internally within the USP, there were 2 drafts published for public comment in Pharmacopeal Forum in 2015 and 2016. Then there was the final version published for 2017 with an erratum, and that has some very significant context as we'll go through. Although the look and feel of this document looks and feels the same as the 2008, there are some significant differences, and you really have to read and understand this to get a good understanding of what's involved. And really from that, you have to understand what is the impact on your laboratory procedures for instrument qualification. So let's have a look and see what are the key requirements. There is a new requirement for user requirements to be documented. Now, the current version or the old version of 1058 only had a requirement for design qualification. You now have to write user requirements. The next thing is that you have to carry out a risk assessment to determine the correct approach for qualification for all your equipment, and that is based upon the fact that all the examples that were present in the 2008 version have actually been deleted and are no longer present. It is dependent upon the intended use of the requirements, of the actual piece of equipment. You determine which group it goes into based on the intended use of that instrument. We've also got more granularity in groups B and C. For example, in group B, there are requirements, or there are subdivisions into straight qualification, qualification with verification that calculations work. So if you use a balance for weight uniformity, that calculation built into the balance has to be verified. And on some pieces of equipment, you have to verify that any user-defined programs actually work, as well as qualifying the piece of equipment. There is also the ability to combine qualification documents, and this is now harmonized with European Union GMP Annex 15, Clause 2.5. In the 2008 version, of 1058, software was looked at as a separate process. In this new version, we have tried to combine equipment or instrument qualification and computer system validation. And the last thing which we'll cover towards the end is that there are changes in the PQ definition and the approach. And it's it's more rigorous than before. Let us look at one of the things at the heart of 1058 is the 4Qs model. And this is the current, or the 2008 version, which essentially I've shown as, in this slide, as a very simple V model, where you have design qualification, installation qualification, and operational qualification. You confirm that the OQ matches the DQ, and then you go on into performance qualification. That's the old version. The new version has some substantial changes, and you'll see this in this slide. There is now, with the erratum, a specific requirement for the laboratory to write its own user requirement specification. That is then used to check that the piece of equipment or instrument that you want to buy matches it, and that's the design qualification. Knowing the intended use and what you want to buy, you carry out a risk assessment to determine how you're going to qualify this particular instrument. The IQ takes all the pieces that you've ordered, installs them, integrates them together, and then you carry out the operational qualification, not against the design qualification, but against your laboratory user requirement specification. Then we go into the PQ, and eventually, after the lifetime has gone through, we go through the retirement phase. So when we come to Part C, we're going to spend a bit more time looking at what this means in practice. With the deletion of the instruments, we have now basically three strategies. With Group A, the strategy for qualification is correct functioning by observation. With Group B, typically we will calibrate. And in Group C, we'll qualify, and where there's software involved, there will also be validation. But what is very important to understand is that the same instrument can be in a number of groups depending upon its intended use. Therefore, whenever you buy a new instrument, you really have to define the intended use of that instrument and determine the group, the USP 1058 group, that it falls into. So it's essential now, with the new version of USP 1058, to develop a good understanding of the changes and their impact. The other thing we need to consider is, where does analytical instrument qualification come in helping ensuring data integrity? Data integrity is obviously the hottest topic, and we need to figure out where analytical instrument qualification comes in helping data integrity within a regulated laboratory. Now, I've been involved with developing a data integrity model. This consists of a foundation and 3 layers. The foundation layer, and this is deliberately called a foundation because I equate this to building a house. If you haven't got good foundations, the house falls down. So the foundation consists of getting the right culture and ethos for data integrity. It covers things like data governance, management leadership, data integrity policies and procedures, training in those procedures, open culture, all of those things. If that's not right, then you've got a problem. But sitting immediately above the foundation layer is analytical instrument qualification and, where appropriate, computer system validation. So level 1, you get the right instrument for the right job. Above that, you get the right analytical procedure for the right job. You have validation and, where appropriate, verification of analytical procedures that are validated for intended use. And at the top of the tree, we have the right right analysis for the right reportable result. The whole point of this layered model is that the requirements are built of the successive layers are built on the layers underneath, and the premise is that those layers work correctly. So this comes to the end of Part A, giving you an overview of the new changes in USP 1058, and I'll hand over to Ali to conduct a polling question. Over to you, Ali.
Thank you, Bob. I would now like to ask our audience to participate in our first polling question. Please click and enter your answers directly on the screen. And here is the first question. Has your laboratory changed qualification to align with the new USP 1058? Yes, no, or not applicable? I'll just repeat that. Has your laboratory changed qualification to align you with new USP 1058? Yes, no, or not applicable? Thank you very much for participating in our first poll, and it's back to the presentation.
Thank you, Ali. And we see that approximately a third have said yes in that poll. I think it's interesting. Historically, when we've done this kind of poll, approximately 2/3 said yes. I think 1058, the nature of it is the more you read it, the more you realize the significance of the changes. So in Part B, I'm going to talk around the role of analytical instrument qualification in data integrity, and I'm going to start with the data quality triangle, shown here. This is directly from USP 1058. It's in both the 2008 and the 2017 version. And what's interesting is if you add a foundation layer and you color code the other layers, it looks like this. And it's very easy to see from this where the four-layer data integrity model comes from, where the green level 3 is the analysis, level 2 is the validation of the analytical method or procedure, the gray level, level 1, is AIQ, and the data governance is the foundation. So this model is a hierarchical model, and you'll see that as this animation builds. Effectively, each level depends on the levels beneath it. All levels are required, and only level 1, the AIQ, is specific to the instrument. AIQ is effectively the scientific soundness in part of the analysis, and the data governance is the cultural foundation for data integrity. And this ties AIQ into the four-layer model. If you are audited by an auditor, the two things they may challenge the integrity of your analytical data on are the data integrity of the work, and that could be because of how the work was performed, how it was documented or recorded, or maybe because of data integrity risks. And people are familiar with the ALCO or PLUS model that regulators have used for their guidance: attributable, legible, contemporaneous, original, and accurate. the scientific soundness may be challenged around the significance of the work. And here I've got an example of a CFR, Code of Federal Regulation code. I think the key things to understand are, is the analytical method appropriate for the samples that have been tested and the analytical decisions that are made on those results? In essence, is the analytical method validated for intended use? Equally, is the instrument suitable for intended use? And to some extent, both of these should be covered in the User Requirement Specification, or URS, indicating the fundamental significance of having a URS for an instrument. For qualification non-compliance data, instances where regulators and auditors have found things they're uncomfortable with, the two main sources are the FDA and the EUDRA GMDP database, which is online. This is essentially the European equivalent to FDA warning letters. With the FDA, we have warning letters, but we also have other FDA data, such as 483 data. You can search warning letters. They're all available online, but the FDA only provide a limited number of 483 reports online, but they do provide significant summaries every year of 483 data and information around 483 citation. Now let's look at some examples. The two here are taken from EUDRA. Report 35325 contains the wording shown. Out of a list of 62 instruments, only 4 were fully qualified. A further 5 instruments had undergone only DQ, IQ, and OQ steps. So significantly, they're picking up on the fact that there was no PQ performed. Another example. Report 35704 says, this included a gross failure of change management, permitting the use of an unqualified HPLC system. So in this instance, the auditor is identifying use of an unqualified system as a gross failure. I think when you review a lot of non-conformance data, the way that the auditor has worded the report gives an indication of their strengths of feelings about the subject. Now, in terms of the FDA and pharmaceutical warning letters, a lot of focus has been placed on chromatography instruments and HPLC instruments in particular. And I'm about to show you a graph that shows the trend. This shows how many warning letters the FDA issued each year that were related to HPLC. It is the analytical technique with the most warning letters, and we see that the trend is upwards, and it focuses on a lot of different aspects of how the instruments have been used. Now, if you take that graph and analyze and review all of those HPLC warning letters and assign a category, why was this HPLC mentioned in this warning letter, we see that the red line associated with data integrity is the most significant one. We see that effectively, in recent years, it's tended to dominate warning letters around HPLC, and that other citations, such as poor method validation or the instruments not being qualified, have tended not to be covered by the FDA. A further example of this dominance is shown here. So the small graph in the top left is just to show the number of warning letters related to data integrity the FDA have issued each year. And you see that there's a significant linear trend. This means that if you want to understand what the FDA are concerned about analytical instrument qualification, you have to look in further detail. You have to look in other data, such as the FDA 483 data. And as part of that, from the FDA summary, every year the FDA issue a summary of 483 observations for that year. And if you analyze the observations associated with CFR 211-160B, which is around scientific soundness of laboratory controls, we get this kind of relationship. So you see, for example, that in 2017, the FDA issued the number of 483s referenced this particular CFR 124 times. Now let's look at some examples. On this slide, I've got 4 FDA examples. In the first one, it says, our inspection revealed discrepancies between the printed chromatograms and the operational qualification protocol for the HPLC system. So this is an example where the FDA are raising concerns about the data integrity of the qualification. The other 3 examples are from 483 reports. In this one it says, during the qualification of HPLC System 10, 4 consecutive tests were performed until a passing result was achieved, again indicating concerns about the data integrity of the qualification. In this example, it says, your firm has not performed performance qualification on the following instruments. And in the actual report, it lists the instruments. So they're looking and picking up the fact that a PQ wasn't performed. In this example, it says that specifically the HPLC, GC, and in the report, it lists instruments were used outside of the calibration range. That means that the qualification didn't cover the range of use. And it's important to understand that in the Code of Federal Regulations, they use the term calibrate rather than qualify. So at this point, I'd like to hand back to Ali for another polling question.
Thank you, Paul. I would now like to ask our audience to participate in our second polling question. Again, please click and enter your answers directly on the screen. Here is the second question. How has your laboratory implemented compliance with USP 1058 using protocols that can be configured to match the URS and range of use, using fixed protocols or not applicable? I will just repeat that. How has your laboratory implemented compliance with USP 1058 using protocols that can be configured to match the URS and range of use, using fixed protocols or not applicable? And thank you very much for participating in our second poll. And it's back again to the speaker.
Hi there, it's Bob again. I see we've got 44% use configured protocols to match, 21% use fixed protocols, and 34% it's not applicable. Okay, let's move forward into part C, looking at how to comply with the new USP 1058 requirements. If you remember from part A, we were looking at this V model, expanded V model for the new USP 1058. And I want to focus really around the heart of the 4Qs model, but also to include the user requirement specification that now is explicitly in 1058, because this equates to 21163 under the GMPs from the FDA, where you have to define intended use of your instrument. So if we expand that little section, you'll see we have a little more detail. And don't worry, I'm not going to go through and start talking about this. This gives you the overview, and I'm going to break this down into a number of successive slides. The most important part is defining what you want the instrument to do, and then comparing it with instruments on offer to select the best one based on performance, cost, and your knowledge of the supplier. That's what it says in USP 1058. However, this is, based on my experience, the worst part of the whole of the 4Qs model, because we don't usually define what we want. We either use the instrument supplier's specification, or we just know what we want and we go out and buy it. And that's where the problems start. If you don't define what you want the system to do in terms of its intended use, any regulatory needs like data integrity, the range of use for the different parameters, any calculations that are carried out, and also the software requirements, you have a major problem because you have not defined your intended use. Once you have that document, you can compare it with what's on the market, select the system that's best for your needs because you have a guide that says, this is what I want. This is the user requirement specification. Don't forget that a URS is a living document. You can change it. But one of the things that you have to do is document how the instrument meets your user requirements. Now, you may say, well, hang on a sec, that's a bit over the top. But actually, that is consistent and identical with Annex 15 in European Union GMP on qualification and validation. And if you look in clauses 3.2 and 3.3, you'll see user requirement specification followed by design qualification. So we have harmonization with USP and EU GMP. If anyone is interested, this month in Spectroscopy magazine, in my Focus on Quality column, this Focus on Quality column is looking at writing user requirement specification, and you'll find it online by the middle of the month, I think. So, how do we write a user requirement specification? Well, we have in USP 1058, it says for commercial instruments, requirements are expected to be minimal. Now it says minimal. That doesn't mean non-existent, written on nothing. It means written and formal, because you're going to test against this, if you remember from that V model at the start of this session. But the question is, how minimal is minimal? And I know there's someone out there thinking, we'll just get the supplier's specification and we'll use that. No. You want to make certain that there is enough information, A, for the selection of the system, and B, to perform the operational qualification. So let's have a look at something very simple for an HPLC pump. If you look at the manufacturer's specification, it has a range of operation that the supplier will support between 0.5 and 10 mils a minute. However, what do you really want? Now, I've put the example down here as 0.5 to 2 mils a minute for conventional chromatography. This is an average. You decide what you want. But the key thing is it is essential to understand how the supplier has measured that specification. What are the conditions they've used and how is it measured? So what I would say is talk to your supplier to understand it. Don't just accept that, that's it. They may have measured it under certain circumstances that you will never be able to reproduce. Talk to your supplier. Okay? So, the one thing that is in 1058 that it is the supplier's responsibility is to ensure that specifications are realistic to help the users select their instruments correctly. After we've selected the instrument based on a number of things, we go forward to writing a purchase order. And you may think, what are we talking about? This is not in USP 1058, but the purchase order gives its approval of the supplier. You get a quotation from the supplier. You'll write a purchase order, and you will have the items delivered. That purchase order and the supplier quotation provides the baseline for your configuration management of that particular instrument. You'll receive the items and check the delivery works okay. You've got all the components, and the supplier's engineer will come in and carry out the installation qualification. It's important to understand that within USP 1058, we have the ability to merge documents together where appropriate. And what I think you're going to be seeing over the course of the next few years is the integration of documents such as the installation qualification and the operational qualification that makes it a lot simpler. You can then provide, or you can then sign off from a laboratory perspective, one document instead of two. And of course, this is also, as I said earlier, consistent with EU GMP Annex 15. So we've got harmonization there. Moving on, let's have a look at the operational qualification and performance qualification. These have to be checked against the overall user requirement specification. So what you must do is check that the OQ matches your URS. If it doesn't, you have a problem. If the OQ doesn't cover that, what are you going to do? And we'll look at that in a slide time. You will carry out checks using calibrated equipment, reference standards, calibrated masses, etc., etc., and you want to make certain that all the components are checked and also a holistic check of the whole instrument, including the software. Make certain that protocols are approved pre-execution and also post-execution, because you are responsible for the work of the service engineer carrying out this qualification work in your laboratories. What you also need to think about is, do we need to include at this point a baseline performance qualification check where we're testing the instrument under actual conditions of use, and we need to check this as a baseline so that we get consistent intended performance going forward into its operational use in the laboratory. And we'll look at that in a little further detail later on. Now, as Ali mentioned in the introduction, I've been involved with the pharmaceutical industry for over 40 years now. And 20 years ago, two colleagues and I wrote a paper on analytical instrument qualification that was published in the Analyst. Well, it's currently called Analytical Instrument Qualification. When we published it 20 years ago, we called it Equipment Qualification. And what we said is the key to deciding if vendor material is useful to your laboratory is to assess the tests carried out against the design specification. i.e. your user requirement specification. If the vendor's material matches all your key parameters, the vendor's material should be suitable for your use. And what that means is you're covered with the operational range in your URS is covered by the OQ, and you show the instrument is fit for use. The problem is that if you just have a static OQ and you are working outside of that operational range, you have a potential problem because that use outside the operational range is not qualified. So you have a choice. If you are using a static OQ, you carry out a gap analysis and work out, are you going to go to a flexible or configurable OQ, or are you going to carry out and do the work, the qualification work, to fill the gap? Then we need to think about linking PQ to the OQ and also to the URS. Now, what it says in the new version of USP 1058 is that performance qualification demonstrates that an instrument consistently performs according to the specifications defined by the user. This reinforces the message for a user requirements specification, which you'll all have, won't you? And is appropriate for its intended use. The PQ verifies the fitness for purpose of the instrument under actual conditions of use, and its continued suitability for intended use is demonstrated through PQ, continued PQ. And the user must define the PQ plans, including test procedures, acceptance criteria, and frequency. So 1058 pushes back onto you, the user, the requirement to define how you're going to carry out PQ. Now, we can have a look in this next slide around Group B instruments and Group C instruments. Now, Group B instruments, simple equipment such as balances, pH meters, pipettes, you can relate through calibration and ongoing checks, you can relate directly back to the URS requirements. So you've got calibrated masses, you've got calibration solutions for pH, and you'll have in your instrument logs the checks against, and you'll be able to plot and map all of those. That's relatively straightforward. It's a direct comparison of the performance of the instrument versus the user requirements specification. In Group C instruments, however, we typically can't do that. We have to use analytical procedures. And so the methods are based on testing and also are indirectly related to the user requirement specification. So what you have to do in developing your test plan for your PQs of Group C instruments, such as chromatographs, is, am I going to base it purely and simply on method-related system suitability tests? You've got to figure out how those SSTs and the criteria and the acceptance criteria relate back to your user requirements. You could also add a very specific holistic PQ test, which could be based on using simple model compounds, simple separations that, again, being method-based, you relate the performance of that test back indirectly to your user requirement specifications. Or alternatively, you have a specific analytical PQ test that is specific to your particular application. So you have some options to consider, but it's becoming a lot more formal and a lot more controlled. And you think, no, this is not right. Actually, this is good analytical science. If you come back to the data integrity model that I was talking about and Paul explained in a lot more detail, you now understand the importance at level 1 of good analytical instrument qualification. You want to assure that the instrument is operating so that the data that you generate, you can trust the numbers. If you can't trust the numbers, what's the point of doing the analysis? But you have to choose the approach that you take to performance qualification. Okay, I'd like to hand back to Ali for another polling question, and then we'll move into the question session. Over to you, Ali.
Okay, thanks again, Bob. I'd now like to ask our audience to participate in our final polling question. And again, please click and enter your answers directly on the screen. Here is the final question. Are you confident that your qualification satisfies new USP 1058 requirements? Yes, unsure, or not applicable? I'll just repeat that. Are you confident that your qualification satisfies new USP 1050A requirements? Yes, unsure, or not available? And thank you for participating in our final poll. And thank you very much for that informative presentation. Before we get started on the question and answer session, I would like to remind our audience how to submit questions. You can submit questions by typing them in the Q&A box, which can be found on the right-hand side of your presentation window, and we'll try to answer as many questions as we can. And the first question, is the risk assessment component of the AIQ strategy a formal process document? And Bob, I don't know if you could answer that first.
Is the risk assessment a formal process?
Is the risk assessment a formal process?
Is the risk assessment component of the AIQ strategy a formal process document?
Yes, is the simple answer. For those that are interested, Chris Burgess and I published a risk assessment based on the new 1058 5 years ago in Focus on Quality in November 2013. That's available on the Spectroscopy Online website, and you can access that and have a look at it.
And Paul, do you have anything to add to that?
Paul, do you have anything?
I think what's important to appreciate is that for consistency of application, it is better to have a formalized process. And the example that Bob's given a reference on is a good foundation for that. You will always reach a point where you have to make some decisions that you are able to justify around any options, whether it's a class, a group B or a group C, and whether or not calculations are included, etc. So you'll always need to reach a point where you have to make decisions that you have to be comfortable defending.
Okay, thank you. And the next question is, do we need to derive a URS for COTS type software containing instrument as well?
Okay, let me answer this one. Firstly, I would define, the question is that COTS is not the best phrase to use. I prefer the GAMP software category. The reason for this is that COTS can be can cover a variety of things. But let us assume that this is commercial off-the-shelf software or configurable off-the-shelf software. So I would equate this to GAMP category 3 and GAMP category 4. So that's commercially available, non-configurable software for category 3, and configurable, commercially available software, GAMP category 4. So do we need to write a URS? Yes, you do. Because of data integrity, this will not be minimal. Again, you'll find more information on the Spectroscopy Online publication that will come out this month in my Focus on Quality column, where we talk about URS, and I give you more examples of that. I'll pass over to Paul.
I think one of the things that Bob and I have realized, the more we've looked at 1058 in detail, the more we've realized the significance of the changes. And in order to help laboratories both understand the significance and also apply 1058 changes in their laboratory, with Bob, Agilent has written a number of white papers dedicated to different aspects of 1058, and those will be available soon, just to add to what Bob said.
Hey, thank you. And the next question is, do I need to perform AIQ for all my analytical instruments?
You want me to take this, Paul?
Yeah.
Okay. Yes and no is probably the easiest way to answer that. If you do a risk assessment and it comes out, the instrument comes out as group A, you don't have to do anything further. Once you go into groups B and C, yes, you do. So it's, in answering the question, it's most things, but not everything. Paul, I don't know if you want to add.
Yeah, I think same as Bob really. By consistently applying a process-based approach that includes risk assessment and includes all of the elements, you then consistently follow the process, but where it's a category or group A, for example, where you verify the operation by observation, it's important that observation occurs at the point you're using the system rather than independent from it. So, for example, you will always examine glassware for chips and marks, etc., before an analyst uses it. You can define a risk assessment that says we don't need to do this because I think historically a lot of companies may have done that. One of the changes in 1058 means you're more likely to have to do it and justify not doing it, whereas in the past companies may have not done it and justified where they need to do it. So there's a subtle but significant change.
Okay, thank you. And the next question is, my chromatography methods include system suitability tests. Do I also need to perform a separate PQ? Hey, Bob, could you take that one?
Yes, I'll take that one. It depends on your risk assessment, whether you just relate to SSTs. What you must do is relate the output of the individual SSTs to the user requirements specification. So, for example, if you are having a method that has a 1 milliminute flow rate, you can relate the retention time, consistency of retention time back to the URS in terms of reproducibility of that flow rate. So, yes, you could, but is that enough? And what you have to do is, where's the plan? Where's the information that you need to justify that you are meeting the user requirement specification and the intended use of that particular instrument? You want to pass on, Paul?
And so nothing further to add to Bob, really. I think, in my experience, it depends fundamentally on what you include as system suitability tests. Historically, lots of companies performed what I would call good chromatography practice, where they had valid, meaningful system suitability tests. I think Lean Sigma may have resulted in what might have been good practice being reduced. So in our experience, we've even written a paper around risk assessment of HPLC that relates to system suitability tests. I think fundamentally, if you only rely on system suitability tests, there is now a risk that the regulator will not accept that as an answer. Very hard to gauge what that risk is, but there is definitely a risk. We're waiting for 483s and warning letters to come through because the update to 1058, the most recent one, happened in December, and we're waiting for that experience of how auditors are applying it to come through the system.
Okay, thank you. And we have another question who wants to know, what about older equipment? Do we have to apply the same level of risk assessments on these older equipment as new equipment? And Bob, can you take that one?
Yeah, I can take that. My advice to you is yes, you don't have to do it all in one go, but you need to start having, this will possibly come under, I think at least you should have the documented assessment of which group it sits in, what are your user requirements, and again, there is the element here of if you have 5 or 10 of the same pieces of equipment, don't write a user requirement specification for them all. If they all work to the same spec, just say they're all you can just have one URS to cover all of those items of equipment. If it changes, then you got a problem. You need to update the URS. But coming back to the risk assessment, I would look at this as a phased approach over time. The risk assessment, if you have a look at the November 2013 paper, is actually relatively straightforward and can be done pretty quickly. I mean, 15 minutes, it is not a great deal of work. And again, if you have multiple copies of the same piece of equipment, then one size in this respect can fit all, providing they all have the same intended use. That's the key. Paul, do you want to add anything?
I just confirm what Bob said, really, so nothing further to add. Great answer.
Okay, and we have another question. For systems such as HPLC supported by Empower, where there are established methods, do we need to have URS as well? And if you could start with that one as well, Bob.
Oh, yes. Well, I would hate to put in a plug for my the second edition of my Validation of Chromatography Data System book at 255,000 words. Do you want the good news or the bad news? Well, actually, there's no good news. Yes, you have to write a user requirement specification, because what is your intended use? And it's not just Empower, it's any chromatography data system, it's any piece of software you use in a regulated environment, including spreadsheets. Okay? Now, with the CDS system, which is category 4, in addition to the URS, or as an associated with it, you need to document the configuration settings, the system policies, and also the access privileges for each of the user groups that you set up. So you cannot assume that I've just done an IQ and an OQ of the chromatography data system software. You have to have a user requirement specification, typically a configuration specification, traceability matrix, because this now comes under Annex 11, Clause 4.4, where you have to have traceability of requirements throughout the life cycle. That gives you a number of things that you have to have. Paul, I didn't know if you want to... No? Okay. I think I've probably killed that one quite nicely.
Okay, thank you. And can we say software validation as per ISPE GAMP is part of AIQ, Bob?
We referenced when we drafted, when Chris Burgess and I drafted the first version of the revision, we did cross-reference the GAMP Good Practice Guide on lab systems. That will work. In some, it's a good approach, and having been involved, both Paul and I having been involved in writing it, support the contents. Yes.
Okay, thank you. Unfortunately, we're out of time, but I'd like to add that the speakers will be able to answer any questions you have in writing, so please do submit questions. But unfortunately, we're out of time. I want to thank the audience for attending and participating in today's event. I'd also like to thank our sponsor, Agilent Technologies, for making today's webcast possible. Concluding today's event, we invite you to participate in a brief survey from our sponsor. The survey will automatically appear on your screen, and we hope that you will participate. As a friendly reminder, please also note the resources provided by our sponsor, which you can locate in the green resource widget located in the dock at the bottom of your presentation window. Today's programme will be available on-demand viewing till April 2019. You will receive an email from LCGC alerting you when this webcast will be available for replay. Please do send this invitation and announcement to any colleagues who may have missed today's live event, and we look forward to seeing you at another webinar soon. See you next time. Goodbye.